Have you seen a ripe banana? And on that small, tiny things that keep flying, is that fly I'm talking about. And these tiny things, they work so intently so that we know how genes are transferred from one generation to another generation. Anything? You thought that it might be interesting to see what this room is about? No? Okay.
Ask you a question. So now we seen the fruit fly and we seen genes and chromosomes. How is the genes passed on? Is it randomly or is it selected somehow?
So his question is that how are the genes passed on? Is it random or is it selected? It is both. Okay. And how many of you know genes? Most of you? So if I say that one gene represents one trait, you understand trait? Any one character. Okay. Let us say what? Eye color. It's not so simple, but I'll try to simplify and tell. And they all are arranged in a very organized structure which we call as chromosome. Chromosome, have you heard? Chromosomes are present in the nucleus is what we, right? So these chromosomes, they are, okay, have you heard about somatic and gametic cells? Have you heard about mitosis, meiosis? Wonderful. No, more than that. Okay, so when meiosis occurs, before the formation of gametes, as we call it. When meiosis occurs, these chromosomes, they go to different cells. With each chromosome will go the genes that chromosome is carrying, right? And there is an event that occurs in meiosis we call recombination. Heard about it? During that time, the pair chromosomes, they cross each other, right? So some of these genes, as he was saying, get randomly distributed to one or the other chromosome. And therefore, at the time of going to these 4 different cells or the 4 different gametes, they will be independently going. And then when an egg and sperm fuse, it is going to give a very different combination of gene. Having said that, this is a random event. At the same time, there is something what we call as linked genes. Okay. So suppose in a chromosome, 2 genes are very close to each other. So even if there is a crossover, the chances are that those 2 genes will be always together. Okay. So sometimes they're also kind of not so random, so they call it linked. So this is what Morgan, you people must have read about Mendelian theories, Mendelian law. Mendel said that law of independent assortment. Morgan added to that. Initially, he was not very convinced that whether Mendel is correct or not. He was not very convinced about chromosomal theory of inheritance. He was not convinced about even Darwin, whatever he proposed. But slowly, did you see that he was emphasizing a lot on the red color and this white color suddenly? He started further researching, he and his group, I should not say he alone, his group started working on looking at this, why this white eye comes. Eventually, they found that it is present, what they found is that it is present in one of the X chromosome. Okay. And because it is is present in X chromosome, though, as you know, the males would be having only single X chromosome. So whether it has Y or it has red, it will express that. But females will have two X chromosomes. So if the red is dominant over white, they will have red. But if there are two whites, then it will have a white one. So with these is slightly complicated, possibly when you people will read about it. When Mendel proposed that, as he said, genes are independently assorted, he further extended that, no, that is not always the case. There can be some population of wild type, there is sudden spontaneously they see a mutant. Further trying to understand that led to a very new concept that unlike what proposed that genes are independent.
I just wanted to ask what causes mutations themselves, other than like an environmental advantage, like a bad mutation, what caused that?
She asked what causes mutation and if there is a bad mutation, what can cause? There are several mutations. How many of you are assault on that DNA? Assault in the form of excessive of radicals, reactive oxygen species, or sometimes because of various environmental process, also naturally, need not be always an assault, it can be also naturally. You have, you know that DNA, it makes its own, it replicates. Do you know that DNA replicates? Yes or no? Have you heard of it? So during that process of replication, there can be certain errors. Okay. So you know that no system is 100% efficient. During that process also, there can be some random errors. And all these changes are generally called, whether it is because of environmental effect, some kind of assault to your genetic material, or a natural process when it is trying to replicate. It will, because when the cell divides, it will also replicate, and it again replicates. Every time the machinery cannot be as efficient. So there are certain random, very random, most of the time harmless changes that occur. And they all are called, these changes are known as, which are mutations. And second is that some of these mutations can be bad. But how? You know that genes are made into, DNA is made into RNA, then it is made into protein, and then protein is the one which functions. Suppose I take an example that protein which we are talking about is an enzyme. It forms a very critical, and it has a very critical role in the metabolism of the cell. Now during that process of replication, that DNA had some mutation. Now the DNA, if it has a mutation, the RNA will also get altered. Hence the protein which will be made will also be different. Now it can happen that because of that change, that enzyme which is formed now doesn't work properly. And that is a very detrimental, or that's a bad mutation as you call it. So immediately that mutation is selected out. That cell will not survive and will not be selected. So only in the cell which had a favorable mutant or which has mutant which is harmless will survive and further propagate. And the cell which is harboring the bad mutation, as you call it, will be eliminated, so that population will slowly die. Does it make sense? This is how the good and the bad mutations are selected.
Why did you always test it on flies?
Sorry?
Why did you always test it on flies?
No, we always do not test it on fly. Fly is one of the, what we call it as, model animal. We do a lot of tests on bacteria. So, bacteria represents the simplest form, and at the same time, it is sufficiently complex that we can say the whatever pathways, chemistry, biochemistry that is conserved in bacteria is also present in our own cells. So from a very, Because it's a very simple system, it becomes very easy to understand. We can manipulate it, we can make them cross. Bacterial generation time is suppose few hours, so we can propagate them. We cannot do it in an organism which takes long time to multiply. We cannot do that. So animal models are used to understand biological problems. Fruit fly is one of such models. There are many other models. We have bacteria or E. coli as a model, Escherichia coli, and you have yeast as a model. We have C. elegans as a model. We have zebrafish as a model. So when we talk about model animals or model organisms, we are talking about a very simple system with the help of which we understand certain basic mechanism, and we know that the same mechanism is also happening in humans. For example, I'll just give you an example. Suppose you have heard about Parkinson's disease, Huntington's disease, lot of neurological problems. So it becomes very difficult if you want to understand the molecular mechanism that is going on during the disease process. To understand that disease process, we go to a model animal that is used as Drosophila or fly, because it has few numbers of genes. Manipulation is easy, and with the help of it, actually many of the disease gene, 75% of the disease causing gene in fly is conserved with us. So if we know that this particular gene is creating a problem in that animal model, we know that we understand what is happening in these complex diseases. So as I said, fruit fly is just one of the model. There are also very simple model, as simple model as bacteria, and you can very confidently say what is happening in bacteria is also happening in the highest or multicellular, single cell of a highest multicellular organisms. So these are models, but there are a number of them.
Why two Y's? If there are two Y's, which gender is on?
Two Y's, okay. So his question is that two X, always people talk about what happens if there are two Y's. Okay, this differs from organism to organism, what we are talking about. If I talk about fruit fly alone, so there is a Y chromosome in fruit fly, but that does not contribute to sex determination in fruit fly. Okay. So the sex determination it takes place with the ratio between autosome, you know what autosome, other genes, and the X chromosome. So if the ratio is balanced, it is a female, as you say. So it will be an abnormal male where you cannot really have a proper determined sex. So 2Y can be detrimental that way, or it will not be very definitive which sex. So there has to be a balance between X and the Y.
And with 3 X chromosomes, is that true?
It's called aberration, and then if an egg carrying 2 X chromosomes fuses with a Y which is carrying an X chromosome, will become 3. So that is called chromosomal aberration. Possibly people will read about that.
I want to know about the crossing over between non-homologous chromosomes.
They are non-homologous crossing over generally takes place when you have repair to do, and sometimes it is aberration.
If you could explain about codominance, as a follow-up.
Codominance is actually slightly different. Codominance, we are talking about the effect of the alleles of the same. Okay, so it is like if there is a white and a black color, there is a possibility of a grey color. But when I talk about linked genes, I am talking about two different genes and not the alleles. Okay, so alleles are the two variants of the same gene or same trait. In fly we have grey body and long wings. People have seen that if they are generally for generations they appear is about codominance and linkage. Okay, there was inside the fly.
The black dot, red eyes dot.
Yeah, it's just see. So when you are talking about fly, you require to have certain markers to identify the what you call it phenotype. Okay. So the black spot, as he said, that people use model animals like fruit fly, and they make transgenic. So that at the very first, after the egg is laid at this first insta, at that time the genetic manipulation is done. So when the organism, it will be the clone of that particular sheep. Making genetic clones all the time. Read about it. It's weak virus, something which cannot cause a disease in us, but can generate an immune response. Yeah, I think. Somebody can ask a priceless question. Somebody so precious that it is priceless. Tell me. Is there any other hand? If you are scratching your head, you can ask the question later to me.
Okay. The disposal of the dead bodies. How they dispose the bodies means it was just shown as a picture, but we don't know the actual process how they disposed.
So there was one thing which they showed in that movie is that they wanted, the family wanted to bury the bodies, but CDC was insisting that it should be cremated. So the reason they were trying to do that is possibly because the virus has come from animals and then it has spread to human. So if it remains in the soil for long, it may go into some some other microorganisms or not only microorganisms, some small animals there or something, and whether it would remain. So the best thing that CDC could think was to completely burn the body and along with that. So what they also showed is that there was a mass grave and there was a disinfectant that was being sprinkled before they were disposing it. So there are ways of disposing, but cremation is one of the way when there is a huge epidemic, people do that, it doesn't spread anymore.
Do is you just have to, all students, you have to go to that web portal and register yourself on that. It's a free registration and you can put in any question regarding science in that and experts will answer your questions. Okay? Plus you can even, if you wish, you can subscribe.
Government College Sakri will join over here at 2 o'clock. We have not made lunch arrangement for this college. Only Father Ragnil Multipurpose High School will go for the lunch because we have done, because previous
See, I got this.
Thank you very much. The teachers, I would like you all to come ahead and collect your certificates, please.
Collect your prize. Good question. And who is that lady? Last question to be asked. Who is that lady? Please come, madam. Who asked the question on the dominant gene, Sandeep? Codominance. Please do come, madam. Collect the prize. Non-homologous chromosome. Somebody raised that question on non-homologous chromosome. Chromosome cross or something like that. Madam is the one? Yeah, please come, madam. Please. It's your privilege. Thanks a lot. Have a nice day.